All right, so I think we've gone through these challenge panels a couple times now, so we know the drill. So very brief introduction to myself. I run an advanced bio-EM platform in the Cancer Center at MIT. I'm in Boston. And I've been doing this for a little over 10 years now. And we offer a variety of workflows of room temperature and cryo versions of TEM, SEM, FIB, light microscopy, and then dozens of variations of everything that links to the next. We have 5 staff members on our team in total. We do almost all assisted collaborative work, and we do work in a chargeback model. And so these are the kinds of questions that were coming up a lot when people were talking about running facilities. And so I wonder if each of you could introduce yourselves, just sort of touching on some of the same things that I mentioned, just so we all sort of have a foundation understanding of where we're coming from. So please.
Me?
Julia.
Okay. My name is Julia Fernanda Rodriguez, and I'm running a facility in Gothenburg in Sweden. I have been in the business for more than 22 years. I think have some experience with managing facilities in different flavors, even when other facilities didn't exist, really. And we have a combination of both. I mean, we have quite a multimodal facility. We are not only based in electron microscopy, we have light microscopy, and we actually have included in the later years, couple of years or 3 years, the imaging, multi-imaging mass spectrometry, and also the nanosim, because the users they need more and more the chemical imaging as well, including into the volume. And that's my background. And my background is in cell and molecular biology.
And so how many people are on your team?
We have 8 people. We have 8 people and 20 machines, different, yeah. It's a big machine, big center if you think about Sweden. That is a smaller country, probably in other countries that are bigger, there are more machines, but that is where we have. And we try to do a combination. When we talk about volume, we talk about volume in everything, not just only in electron microscopy. We sort of go from one system to another, and the correlative multimodal imaging is actually our strength.
And do you use a chargeback?
Yes, we charge for using the systems.
Okay, thank you.
Thank you.
All right.
Yes, so my name is Maria Vancová, and I am head of the laboratory for my.
Can you hear her?
Can you hear me? No?
I have a strong voice.
I might put it a little higher, if you don't mind.
Okay. It's better now? Okay. Thank you. So once again, my name is Maria Vancová. I'm head of electron microscopy core facility in České Budějovice, Czech Republic. And I'm head for only 4 years, so not long. However, I am in the field more than 20 years. And our facility provides just very broad range of different technologies, transmission electron microscopy, room temperature, cryo, scanning electron microscopy, cryo, room temperature. We do serial phase scanning electron microscopy, array tomography, and we provide just a service from the, let's say, planning, help with the sample preparation, imaging, and also image segmentation, and also interpretation of results and so on. In total, there is 11 persons. However, not all are involved in the service. So for services working 6 persons. So that's it.
And you use a chargeback?
Yeah.
Hi, my name is Alice Liang. I'm the director of the New York University School of Medicine of a microscopy laboratory. I started this course in 2005. My background is cell biology, so I'm more towards an EM person. But currently, we are, beside the cryo-EM, we do all the optical microscopy and traditional any other EM work. And for the light microscopy side, it's mainly for training, the instrument maintenance. For the EM side, is everything on us, basically from sample preparation to image analysis, image presentation method, writing those stuff. And we have total 6 persons, 2.5 on light microscopy side, 2.5 on the EM side. But in total, we have 16 light microscope and 2 TEM and 1 zero block phase SEM. And we also run on chargeback.
Our facility feels so small in comparison. This is exciting to talk to all three of you. So in all the different conferences that I go to, all the different meetings, getting to interact with colleagues, a number of challenges always tend to come up. Doesn't matter what environment we're in. And so I made a list of some of the most frequent things that I hear about. And the first one I wanted to touch on, and it may be a little bit different regionally. In fact, I would actually just comment on this, that I think I have found that our communities are so diverse that different approaches are really appropriate for some communities more than others. So, and also terminology may be different. So I sort of recognize in advance that we may refer to each other differently. So no offense is meant anywhere. But one of the things that always comes up in my area is the idea that EM has oftentimes been called a service, and approaching that against the other idea of EM being a science. And you'll hear Lucy Collinson, I know, talks about this a lot. I hear Ru Cheng at the NCI talk about this. And so I really wanted to bring up this topic for everyone of EM as a science, not as a service, and just hear your thoughts on that and whether this is important in your world. Do you come up with this as a challenge?
When you talk about science, you talk about collaboration base. Is that what you mean exactly?
Yeah, I mean,
just to be sure
yes,
that I
yes,
understand
so that
the terminology.
That it's not a plug and play. That you can't just, if one person is sick that day, you can't just hand it over to another person and let them put in the instrument. And so, whereas oftentimes services are thought of as, oh, anyone can do it, you just happen to be the people that have been hired, versus understanding that this is actually real science. It may require iterations. It may be exploratory when they're coming to work with you. And maybe this isn't the case.
Yeah. You don't have it on.
Sorry, once again. I said that times ago we did mainly illustrative images or something, and now we do more complex studies, needs different correlative approaches, combine different techniques, and that's something that we started to be members of different teams, at least at our institutes, which maybe we're talking about that. So we are still calling salaries. However, we are part of many teams currently, and we help them because they don't understand too much. They are lost in all protocols, technologies. We can help them if they have some problems. We can help them to find the right technology, right protocol. So they need us as a members. So to be this answer.
So totally agree with you. To add on that, I think we in NYU basically we change everywhere. We don't call our core facility, we call the lab. Just because we don't simply think we are doing the service alone, because currently, as everybody's doing their own research, not many people really know the microscopy. So how for follow up their project, usually they talk with us is about how do I resolve this question. So we have to design from the lab microscopy to EM, or from the cryo-EM to the lab microscopy, whatever workflow we can give to them. And it's purely education at the beginning and to the end. You have to explain to them what the structure look like. Well, what kind of technology can solve what kind of question. That's from that one side. I think everybody doing very similar thing. On the other hand, how do you put a new technology to the school? That's also our job. basically, there's like a lot of PI has their own stuff they're interested, but we are the person always ahead of time, try to think about what is the new technology we want to bring to the school. Then we try to talk with several PI and try to get some demo or check some primary data and say, see, that's what we can do for your project. So then we grant or at the end we got funding from school either way, but I think this is like a purely collaboration project kind of thing. So currently our school is always put us on the, whenever new PI come to NYU as a recruitment processing, we are at like a second round interview. Basically, they need to talk with us, what they can do, what we can offer, what we can do continuously. So purely based on that, I think it's really, we are not, I totally agree with you. We're not really service, we're doing part of the research.
So this is actually a conversation I was having last night of what do we call ourselves? And I've gotten really frustrated sometimes calling myself a core facility. And so I will sometimes call myself a lab. And I've actually now started, I've now decided we're an innovation platform.
I still call a core facility because I think we are the core. And then the different, this is a bit different view, how you expand from the local, regional, national, international, and that's how we work. And then I always think that we are at the core. The name is irrelevant. In the way we work more in the facility, and as I say, we are divided in teams that do light microscopy more, EM more, and image processing analysis in the way that we go for end-to-end data management and also including the sample preparation. We sort of divide the situation in service and innovation or innovative job or development job, where we have to develop new methods, new technology. And when I say new tools of technology, I'm including as well all the processing, all the analysis tools that we have there. Then there is these differences in between what is proper service, more routine, and that's the way that I training in the microscope, mainly for the light microscopy. But for vEM, I followed many of the ways that I have been working for 15 years with the light microscopy in training as well. People, there are scientists, they have to know, the majority are PhDs and postdocs, they need to know what they have in their hands. And I'm not afraid that they're using the machines, of course, in a different way, the training we have. And then start working with us. And we don't do that job only us. It's not only my team will do all the job. In light microscopy, definitely not. We training, that's why we call them users. We training them, they use the instruments, and we support them in the long run. If they need a development, a stronger development because the project need that, then it's a contribution. And then my personal will always go in the papers probably because they have contributed more than just to routinely training people in using the instruments. In the electron microscopy, there is a bit more development coming in every single step where the way we're thinking have to be a little bit different there, but not too different compared to where we do for the light microscopy. We always say, okay, the first method we're developing, that's innovation. When we have that method ready, it becomes a routine method. Some of them can become easily, some of them cannot, because we have really different flavors of light microscopies. We can run from the light sheet, multiphoton, super resolution, conventional, confocal, scanning electron microscope, transmission electron microscope, including chemical imaging. The guys that work with me, they have to be able to jump in between the microscopes. All of them have a PhD background and expertise that not only is the microscopy, some of them are biotechnologists and do 3D printing.
So your team, you mean?
The team. And then we included always constantly the user in that interface. So they have to work with us constantly.
Let me ask, how frequently, what percentage do you think in these more advanced workflows and protocols are you training users versus you are collaborating with them and doing the work yourselves, your team? And I would ask this to all of you.
Did I start? Well, we have around 20%, and I have this 20%.
Where you train?
For the helping them in the method, in the collaboration.
Okay.
In fact, we have that. And we are lucky in Sweden as well that they have fellowships that can run for 5 years. It's like a project in where we call method development research infrastructure fellow. And in this case, these fellows have to be part of a research infrastructure core facility, and we do method development or technology development. And then you already
I like
have a
that option.
percentage. And a percentage of that, where my staff that have that possibility.
Okay.
We have training also. We have courses just to learn the basic methods and how to operate with microscope, camera, and so on. However, I have to say that although we are training, we are doing more and more. Because for them, especially if they do one project in a year, it's not, I think, reasonable to learn the cutting. Because they or just learn some very difficult serial book phase came through microscopic, you know, how to set everything, because they don't know after 1 year is where, and still need a little bit of adjustment to adjust the parameters and so on. The same sometimes is with the segmentation. We learn them, however, moreover, we find them to find the good tool or just for their purpose, and then train them how to use it and to do it for themselves. So it was also changed, I have to say, in years. Still less training, basic things like cutting, for example. I also had a visitor that came to me and say me, "Oh, you are cutting?" I thought, "Yes." "Oh, I'm so glad." So that's the situation.
Alice, what about you?
Yeah, I think maybe similar to yours. Basically, for optical microscopy side, light microscopy is training. And basically, assist use of training, and then as all user. That's why 90% of the population come from light microscopy. We don't have authorship. It's just because after training, then they start to use it, they forgot about you. For the EM, it's a totally different story.
So then, yeah, what's the EM story?
EM story is we do all the sample prep, right? But then the training will only do the TEM. And the TEM imaging, those you can train. But like the volume EM things, no one wants to be trained. We don't want anybody to touch the serial block phase. So this is purely on us. So if we involve a lot, then we will have authorship. If not, then sometimes there's 50% of the chance. Maybe it's like acknowledgement for anything is required, but they don't always acknowledge you for sure. So I think it's similar to all the other facilities. And for the image analysis part, I think I know there's a lot of software around, but we only have a very limited stuff. So we have the commercial available software, which we use. We at the beginning we do Amira and the Dragonfly. Now we focus on Dragonfly because it's easy to use and easy for training. And basically we do training, and the people will do segmentation by themselves. We'll train them from beginning to the end to help all those processing.
We have almost entirely assisted collaborative. Very few of our community is focused on EM. Most of the people in our community, EM is one little, very important, otherwise you wouldn't be paying for it, part, but usually EM is not the focus. And so we do almost all assisted. And I just lost the question that I wanted to go to next.
We're waiting.
But I guess I see that this is different than what perhaps the single particle cryo-EM community has. And I'm curious if the audience has thoughts or feelings about this, because it seems like some communities expect it to be train. But when it comes to how are you actually going to get any work done, it's just not possible with some of these more advanced workflows when this is only one little part of a project, or when they might have to touch 6 different instruments and you don't want them broken. Is this something that comes up with conflict in any ways?
For the cryo screening for the particle, we have come up with a national strategy in Sweden. They call the cryo screening network. You have the platforms, the big ones with the big titans and big machines in Stockholm and Umeå.
Single particle world.
A single particle mainly, but also it will go as well now to the cryotomography. But it started with the screening, And then we have sites in Lund, in Gothenburg, where I am, and in Lidköping, where we have people experts. I have one expert in my lab where they do. I mean, we're helping with all the preparations, helping not with the purification, but with the plunge freezing. We can check in our microscope, but we also send the samples, the grids, to Stockholm, and Jens by remote is running the cryo-EM in Stockholm or Umeå. They are able, Thermo Fisher have done a good job in doing that, and we can actually run in that way. And that means we don't need to have the big machines. We don't need to spend the money in the services. We send it to Stockholm. They put it in their Arctica. We take the screening, the 12 grids or whatever they have in their autoloaders. If everything is working fine, we tell them, put this please in the Titan for collection. And this is a way that the country have as well helped to use much better the scarce funding we have. They found it's like a synchrotron work. You have the big place and you send everything there, but you can run it from home. He's actually running the system remotely from our side in the way that we can remove the overload for the people working in the sites in Stockholm and Umeå. This is the way we did for the particle you asked before.
I have watched some of the national centers come up in the US where they're starting to go in this direction. And I see them try to differentiate the kinds of research or techniques that will be allowed within some of these initiatives. And I find it a little concerning because I think it's sometimes difficult to separate a very specific technique such as some of these volume imaging that may lead to tomography away from all of the other advanced bio-EM techniques. And because we use all of these techniques to get to the final product, and maybe the final end is sometimes a volume, maybe it's not, but many techniques are necessary. Do you find in your spaces that you need to rely on many different kinds of workflows and approaches to work through a project, or is it usually more streamlined if somebody comes in, they know they need one type or another? I see your head shaking.
Basically, like 90% of people come to me, they don't know what technology you're going to use. I said, they only present to you what they want to resolve. So that's why we come up with different strategy, right? And then lab microscopy, like a few people, they know what kind of what type of microscope they want. But for EM, basically, they just tell you, I want to resolve this structure. I want to see how mitochondria look. I want to see whatever. So you design them, you tell them, if you want to see this, I can give you this technology. If you want to see, we can do that. Or from here, you can go here. I give you a different price, what you want to do. So basically, that's the choice. And we also, even though I'm not running cryo-EM core, by the way, I have a very close relationship with cryo-EM core. So things from us can go to cryo-EM core too. So we have a very, like a lunch every month, core director lunch. So basically everybody knows who's doing what. So in turn back to the national facility, basically New York City, that's small in Manhattan, right? We do have a New York Structural Biology Center, which is a national fund. And they have, I think, 12 Titan Cryo already. But so all the affiliated institutions, for example, NYU, Columbia, Cornell, Rockefeller, they all have their own cryo-EM core. It's just because simply you cannot really get enough time for the usage, even if they have a lot. Also, the specific question you ask for, it's pretty difficult for them to really work for you. So this is a category you can use for scan, but mainly for data collecting, people also do here. Also in there, they mainly doing cryo-EM, but they don't do anything regarding resin embedding or the others. Often they send people to me, says, you need to help them with the sample prep part for this or for that. I think that this is all very collaborative situation. All the technology currently all goes streamlined to us, and we try to go whatever place. Occasionally, you have one project. People say, give you a paper. I want to do this. then you can tell them how you can achieve this, or it's almost impossible to do that. So that's not really popular, but you do have people also have expert come to you, say, I have done whatever things before. Then you learn from them, and then you can add up the new technology.
Yes. Did you have anything you wanted to add? Or?
No, I completely agree.
I have something to add there in
Okay.
terms of the volume EM or more advanced technologies that you mix, because we do a lot of correlative. In our case, it becomes volume correlative. We have two populations of users. There are people that they don't know anything about volume EM. They know more about cryo because there's a lot of people and it's fashion, it's a lot of, but the volume EM they are, and then we have the other group that they really know, and they really know what they want to do. They don't know how to do it because they are not an experts in sample prep or in the processing. The majority of the volume EM we're working mainly with a rate tomography. The ones we're doing, it's a pure collaborative. We are sitting in Gothenburg and the user is in Karolinska in Stockholm, and we are actually as well collaborating with someone in Spain or someone in Austria, depends on the projects. So sometimes we really need help from many places in ways to actually make it more collaborative or helping each other. Sometimes if someone is very good in preparing the sample, they do the prep, we do the imaging and the acquisition and the processing and the analysis, and we're discussing. And we have been using WebNosos, I don't know if Norman is here, as a very nice tool, collaborative tool that allow us to share the images. The user is annotating. That's the investigator that they don't know anything, but they want to do volume EM, that's clear. And they are the ones annotating. And then we have a real, and that's the beauty, I think, the fact that we can send when we're working in room temperature EM, because the sample can go anywhere comparing with other samples. And I find that comparing to like microscopy, that sometimes you really need to be close to the lab that is growing the cells. Is a beauty that allow what I love, that is the collaborative work, and because it's amazing to sit with all these people and discuss, because a lot of science come together.
So the word collaborative and collaborations and multiple teams with different expertise keeps coming up, and I think it comes up throughout the entire forum. Yes?
Sorry to interrupt.
Not at all.
I had a question based on something that was said a couple of comments ago. So we talked a little bit about how when users approach a facility, sometimes they know exactly what they want, but a lot of times they don't. And so I guess, how do we make that easier for them when they're approaching? Like, how do we bring them in to a brand new area and kind of help to temper their expectations, right? Manage them appropriately, but also get them to provide like the appropriate information so that we can, end up with something useful for them. And I think the other part of this is, a couple of people now have talked about how they might go from one institute to another to collect data, depending on what the goal is, right? And in my experience, those transitions from one institute to the next can be the most difficult parts of the projects, just in terms of aligning sample preparation, data transfer, data management, getting access to the microscope, all of those little, sneaky parts of the process. So how do we make that easier from your
First,
perspective?
you say something very important, that is the expectation of the user. I think one problem with this kind of long projects, include volume EM or correlative approach or multimodality, is that they have wrong expectations and unrealistic expectations, especially in time, for every process, because they don't know how much take to prepare sample or to acquiring images at the microscope. That this is something that we are working in the facility, and we will want to include more vendors to increase this time-consuming steps and automatize the automated microscopes. We have to make them smart. But I think the expectations is one of the problems in terms of the users. And to work collaborative, it takes time, you are right. But at some point we have to do it, because it will be less money, funding money. And this is something that we discussed here. And we will have to collaborate and cooperate. And the people, because they have the same problem than you, they will not get that. They will be very open, or at least this is where we experience in Sweden. Obviously, we have a national commitment and help from the research counseling and from the ministry itself. But people start to understand that if actually we're working in that way, we are much more effective. It took a couple of years to make the cryo screening, for example, working in the way we do now, but now that is the way. We tell that to our users and they are happy. They're, oh shit, this is great. I want to do that. I didn't think it was so easy to do it. I think, and this is beauty, when you actually tell them and you have solved the problem, but it's not straightforward. Yeah, that I agree with you.
Marie, how do you onboard? Was there a comment?
Yeah, so I was just thinking, could we start recording more the number of hours that each process takes? Because I know that in some of the big papers, when it takes 3 years worth of people hours to make a huge reconstruction, that makes sense. It shocks people. It shows you what's involved. But we do a lot of other processes and we record the hours because we charge for the number of hours that we do. So the information is there. It's metadata that we could put with the volume stacks. And then people could know when they come in, if you want this amount of data to come out at the end, you're going to have to put in 50, 100, 150
I like
hours
that
worth
idea.
of work.
Yeah.
Fantastic. I mean, I think everything that give a realistic number to people that they understand and they know what it is, and they don't think that it's not us, that we don't deliver it, is actually is a fact. It's the way that
Yes.
this kind of
So having a something that goes along with it took this many hours to achieve the data that is presented here. I think that's actually a great idea. Right behind you, John.
Yeah, I found them by far, going back to Michelle's comment, I found by far the easiest method of figuring out what is in the head of a prospective user is getting them to send you a paper figure.
Absolutely.
They have got that information from somewhere, and if you can figure out what paper they've read, you can find out relatively quickly what they actually have in their head and how feasible it is.
Yes, I think so. Marie, how do you?
I would maybe mention, and it was said during some other discussion, that preliminary results are also important, just to test a little bit short data
Yes.
set. And look, it's okay for you, it would be good for segmentation and so on. Also helps, or for labeling just positive controls.
I mean, we do pilots. We do pilots for the, I mean, we don't go with these big things. And especially in the processing,
Always
we need
good
to
to
have
pilot
small
some
things.
things, yes. Alice, what is the process for onboarding? And in this question, I will just remember that during the sample prep conversations, I think maybe Rob mentioned, frequently when a new user comes in, it means an hour to 2 hour conversation with that person. And so I think that may be relevant to this question.
Yeah, that's always the first consultation. Some core facilities charge for the consultation, but we think that's part for free for sure. Besides that part, how the other part is charged.
Yes.
Because I think this is so important that you learn from the project, and some project never did it before. It's also your first attempt, right? So this is, I always have my introduction with all different kind of work we did as an example. What kind of technology we can provide to you, and use the relevant laboratory result as a result. If you want to see this, that's how we did for this lab. So then they have an idea of what we can achieve. And then I think we go deeper to talk about the real, how we should go. So basically the conversation is always 30 minutes to 1 hour. Majority of the time is 30 minutes. And this is the way, basically, the good part of that in the first consultation part, majority of the time is not only PhD and postdoc. The PI is always involved.
That's great.
So after this part, then the PI is out. So this is, I think this is a very good. They know because when they understand, then they are willing to pay. Then sometimes if they don't understand, they always think, we have to pay this much. Then you tell them this is how much hour we charged, and we spend much more than this. So then they understand. So that's current our workflow. And for certain experiment, we don't know which method will work better. We do all three of them, but whichever work we charge for that. The rest of the two we don't charge. So that's I think maybe the beneficial rule. People can give you a little bit of smile on this part, because if it doesn't work the experiment, you charge them. They're very unhappy. So that's why even though we charged, but we keep talking with school, cannot really get 100% chargeback revenue. And ideally it's 50%, but currently our goal is 60-65%, and we always over that. I always think we should use the lab microscopy to compensate the EM part, because the lab microscopy after training is people using, right? You should generate revenue. But now in our core, it's always half and half, basically, at the end is every revenue. That's was how we come out with.
So it sounds like if people recognize that EM is not just a rote service, like going in and having your tires rotated, that they will be more tolerant of data and still paying for the work when the data doesn't come out of it. I wanted to touch a little bit on intellectual contribution and the recognition of your intellectual contribution and your staff's intellectual contribution, which is always like it comes down to authorships. And in our particular community, very few people actually focus on EM. They know very little about EM. And so I run into situations where somebody will say, oh, you know, EM's not the focus of this huge paper. So they are reluctant to include the relevant people as authors. And so I wonder what strategies you use to protect your staff, to protect yourselves, so that you are still intellectually recognized for your contributions, because no one is going to pay this money to put a figure in a paper if it's not necessary. So if it's necessary, then I think your contributions should be recognized. Anyone want to take a stab at this?
During consultation, we already discussed that. It's a little bit what is the, how much they want the contribution. If they have experts in the group, in their groups in the lab, we can just have to help to get access to the instruments or to the labs to do the prep. But this is not the case. The majority of the time, they will need us quite a lot. In the beginning, 22 years ago, it was a bit more complicated to get that, even to acknowledge, because we have fees and they suppose we pay, even if it's extremely subvention, and you have to say you pay your postdoc and PhD and you put them in the paper anyway, then I think you have to do the same here. But during these years, at least in Sweden, they start to recognize and contribute. My team are all PhDs, except our last computer scientists, that they are not. And that means that the contribution they give is going beyond to just show them how to press buttons in an instrument.
That's great, Maria. I have a few more things, so I'm going to start cutting people off.
Yeah, that's right. That's all right.
Yeah, I agree with you. To discuss info, we start. And if somebody don't want to include us, we simply let him work as a user.
I like that.
No, I mean, that's purely base by base. But I think that's something we have very strong at one point says we should be including in the authorship. But, you know, my advisor committee, who is all PIs, they have a very good point. I think I can take that, because they said, you know, conceptually, if you change the theme of the paper or at the beginning of the design of the paper, that you will be part of the authorship. Otherwise, you have too many people involved, especially currently, each paper, the genomic core, proteomic core, so all proprietary stuff will be like that. So well, now,
but should a core, well, first of all, what does it matter? And then second of all, just because it's a core doesn't necessarily mean they're intellectually contributing. Yeah. And Michelle.
Yeah, I was just going to broaden even beyond this, because from a software development perspective, and also from like just an algorithm development or use of analysis or whatnot, it's the same issue, right? A lot of times, in my experience, we will have someone approach us with a biological problem, we'll be asked to do something, and we will develop brand new tools and approaches and like really innovative solutions. And then my team will get put third, fourth, fifth author on the big Nature paper. And so that is intellectual contribution. They're being acknowledged, but I would argue they're not being appropriately acknowledged. And this is, I think, the balance that's really difficult to find, because like that Nature paper would not have been possible like to that level without the tools that were developed, without the sample prep, without a lot of the other parts of that process. But because the focus of it is the biological outcome, the people who did all of those things get the third, fourth, fifth authorship positions. And then if you think about it, right, I have 5, 6 projects like that. That means every single time I don't have a first author paper right then. I have 5 or 6 third, fourth, fifth author papers, which then is detrimental to that person's career trajectory, right? And so I think that there's kind of a larger scale problem here, and it goes beyond just the intellectual contribution, but somehow actually appropriate intellectual contribution.
I like that comment. Are there any other thoughts? I don't want to be leaving the audience out. Yeah, Kirk.
So, I mean, this is like the core facility problem over time. It's always been there. But one of the things we've done is there's 2 things. One thing that I do insist on my users is we have a research resource ID that's unique. So every single publication, they get language that has this unique number that I can go into Google Scholar and pull, because half of my battle is to go to my boss and actually know that they published something, because a lot of times they'll take their picture and it shows up. So that's number 1. Number 2 is I always put the funding agency in there as part of the linkage. So if they use a particular equipment, funding agencies will do this as well. So I need to go in and come back and say 25 papers. Well, that's a good outcome, but you know, a lot of publications. The third thing is my experience sometimes is I don't actually try to grab onto every paper. Sometimes you need to look at the quality of the paper, because they're sometimes putting your core facility on to add credibility to it, to be honest with you.
That's a good point.
And when I look at the rest of their science and I have questions, if they don't answer them, then I say, well, this is more appropriate. So I mean, there's a very politically nice way to say that, but also you have to be a little bit careful. The fortunate thing now is that when journals are coming out and saying, what was your contribution, you can define that. But if you get into a few papers that are just a little bit, you know, lower quality, then there's also some risk to reputation. So I just wanted to make that out there as something to think about.
That's a great thing. I'm glad you mentioned that, Kirk. I wanted to make sure we had an opportunity to touch on career development and career paths, not only for core directors, but also for our staff. And I feel like this is something that comes up frequently. And I would be curious to hear how this is approached, if you feel like you have career mobility, if you don't, what steps are being taken, or what support could be provided from the greater community so that this can be better addressed. And I'm going to start with you just because you're on that side.
Okay. So basically we have NYU has two different systems, right? We are all in the research track, and the research track in the core facilities setting core director, it has academic title, but all the rest of them are staff, right? Then the staff has 2 different categories. One is technician series, another one is a scientist series. Depending on what's your experience, you can go from real basic to the senior to the principal. Then you continue can grow, become a lab manager or the director. And so it really depends on, for my staff, anybody come here, and then we do have the EM and lab microscopy focus. Depends on what is your strength, what do you want to do. And we give you all basic training. Then you decide what is the your major thing you like to develop.
Can I add a question in there?
Yeah.
Are there internal in-rank, I don't know what they're
In-rank,
called everywhere,
yeah.
in-rank promotions,
Yeah.
where maybe you're still the director, but you had some sort of an internal promotion?
Yes.
Okay.
So because, for example, I'm affiliated with cell biology department. You can go with assistant director, assistant professor, associate professor, and the full professor. So that's for the director. And for the core staff, that's different. So that's now you don't have that change. Then the core staff promotion all
rely on me, basically. My recommendations that this one need to be promoted to whatever. Every opponent year, who's doing what. Every year we have this focus and review, and you can give a review and you talk and you see how you want to go. Also, people want to go to a little bit higher education. For example, they come here EM as a technician, they're under degree. I always encourage them to go to master. They're doing part-time master, and NYU can be free because they are staff here. And then that's another thing they can go, and then they can decide if they want to anything new they want to learn.
So these are for people who don't have higher level degrees yet.
Yeah.
Marie.
I know maybe I see maybe the what is missing. It's the training courses for technicians. They be a little bit help between the lab that we know that if there is somebody else who needs the training for, I don't know, array tomography thing, then he will come to our laboratory for couple of days and check out how we are doing and so on. And now it was nice job shadowing a call from a global imaging and global bioimaging, which maybe also help to just share technicians some just to gain some new knowledges, experiences, and so on of them. So that's good because we can, as a scientist, we can travel, we can attend different conferences, workshops, and so on. However, for technicians, this is at least in our country.
So for people who are already scientists and have PhDs in advanced level,
they can.
Do they have career paths or
Not in the core facility?
No.
See, I think that's a huge problem. Definitely, I see that there are more ways for people without their highest terminal degree to have promotional opportunities. But for, not saying it's perfect, sure, it depends.
No,
but
I mean, I'm the head and that's it. I mean, this is a topic that is extremely important for me. I'm part of 2 European projects that are running in parallel right now, Retrain Plus and Arise, that is actually touching that. But I think we have to make here a distinction between career progression and career path. I mean, the career progression, we don't have, there is some institutes in Europe, some universities in Europe as well, especially in UK with a technician commitment. In Sweden, we start to do that where you have a proper career progression in the core facility on the research infrastructure in parallel with the academic.
You do.
Including technicians and you have technicians, administrators, and people that want to have a leadership. Not everybody wants to be a leader. Not everybody wants to be a head. And they can jump in between. This is not yet designed in Sweden, but we are discussing, and we have a proposal right now on our rectors and also at the national level in how we want to do that in the core facilities. But this is not something new. It's not something Sweden. I mean, there is institutes, EMBL, VIB, some institutes, research institutes that they have created proper career paths for career progression for. Another story is the career path and the education. And that's the program, the Retrain Plus program is actually doing that, trying to do that, to create a proper career and to have the new generations of people working in core facilities. It means that you start to explain people about research infrastructures.
Is there a question? Please.
It's more of a comment. So I work just down the road at EBIC, which is the national facility for cryo-EM in the UK. So we have, people with PhDs who are beamline scientists who also help users, but they also, all of our staff as a percentage have their own time for research. So they have a career development path in they can go from beamline scientist, senior beamline scientist, principal scientist. So there's that path, but they also have the chance to do their own research, go for their own grants and things like this. So there are ways to do this. But one of the things with that is that we are probably better staffed than most core facilities. So this is where you need to have, staff level to do that. Because, if you've got 20 instruments and 8 people, well, they're going to be spending a lot of time they're not going to have the time to do a lot of development for themselves, right?
So you're talking about there's development that is an option in terms of people doing their own independent research.
It's not an option. It's they have the
They
opportunity
have
to
an
do that.
opportunity. I guess I'm wondering, I will sometimes talk to administrators and institutions who are very well-meaning, but they're just relatively uneducated and about what probably everyone in this room does. And I have had them say to my face when I'm talking about this, well, Abigail, at some point, the job is the job.
What do they mean with that exactly?
That's a very good question.
Yeah.
And I think what they mean, because I did ask them, what do you mean? And they say, well, you know, you come in, you take your samples, you put them in the instrument, and that's your job. And I think this person thinks that once you've had 2 years of experience, you're no better off with 5 years or 10 years of experience. And they don't appreciate how much everyone who's working in this field is constantly learning. They're constantly gaining new skills, even to the point where one of my staff who does cryo-TEM, not even single particle, we're talking about plunge freezing, cryo-TEM of nanoparticles. He's working with different samples every year, and they don't realize this. And so they think the job is the job. And I really feel strongly that there needs to be some sort of promotion that recognizes how much experience one has, even if they still haven't really changed their job.
But that's what I mean with career path, because there you have to explain the requirements. One thing is the progression and how you go in whatever is the decision they take in your institution. Another story is the career path. What is the education? What do you need to know? What is the profile of a person working? And I agree with you, even if you do that all the time, you also know you are doing service provision to improve your service. It's an innovation. It's not only making machines and innovation. How do I improve the service? How I improve the way that I deal with my users? And this is the extra knowledge that no one have except us. Mm-hmm. And that has to be in these career path and requirements that the people need to know, the budgeting, even if they don't do the life cycle of the facilities, things like that, I think is important.
Yeah, I do agree with you. I think career path is important. That's why you need to have a different degree of the scientist, right? And
well,
I
think
for the manager,
when you say degree, do you mean their educational degree?
No.
Oh,
like a staff scientist, a principal scientist, this kind of thing. Then at the end, you can promote to become manager, assistant director.
But that's a leadership thing, and that's often a very different thing.
Go with your salary and with everything, with the people recognition. It's all along the same.
I guess I wanted to talk more about the difference between leadership paths and technical paths, but I also want to get to this question, and then I want to get to uploading data in repositories. So, boom, boom.
I think it was him first.
Oops, sorry.
No, yeah.
Right. Not much a question, but a comment that in Glasgow, looking for a career path for technical specialists, we created that career path specifically within the university, away from only a researcher or your academic, in a way that actually the technical staff can get promoted up to a professional level. So they created that in a way that we can be evaluated not only on the number of papers, because as a facility, it's hard sometimes to be acknowledged on that and to show, I contribute to X amount of papers. And although I was not acknowledged, but I work on that. So they see how many projects we work in a year, or how many different projects or collaborations we have. So that's a different level of evaluation on our jobs.
At Glasgow?
Yeah.
Okay. We might need this uploaded for everyone to see as a model.
You have a Liverpool.
Okay. One of the problems is also that this is partially nationally regulated, because I know within the Netherlands, we have these promotional lines that are also increasing with monetary, your salary just increases with each promotion you get, but it's also very strict on a job or function description. So for me, I've been at the top of what I can get to for the last 5 years. By now.
And that's the problem.
Well, for me, it doesn't really matter that I do you not get more money? Because I'm pretty
You shouldn't
okay
say
off.
that. Sorry to point at you.
Sorry, more money is nice, but I don't necessarily need it at this point. But I also can't get any higher up because the first thing I need to do is get in money. And with my track record, that's physically impossible within the Netherlands. But there's also no way to say, okay, but I have this and this and this and this layers of experience and all these different techniques that I can do, which would make me more than a level 4 science person or whatever.
I think that this idea of a career path is something that I would, in just development, I hope that can get picked up more. I think there's a question back there. And
Yeah,
then-
I just wanted to follow up on what Leandro was saying. We've also got a technical career path with promotion opportunities at the University of Warwick, and I believe not represented in the room, also at Liverpool University.
Yeah.
And
Great.
this has all come out of the technician commitment, which a lot of the universities in the UK are signed up for. If your university is signed up for this and hasn't done anything about it, go prod them.
That's great. Thank
If
you.
not, the technician commitment will do.
Okay, two very quick ones, and then I want to at least take 2 minutes to touch on data uploading.
Want to tell about a bad example is Manchester. It has a technical review for 6 years, starting over and over again. Nobody's promoted. Nobody knows how things are going on, and they're still in technical commitment. And
Problem.
Yeah.
I think
whatever
that there
you
should
do,
be.
you prod them, you nudge them.
We should have a bigger conversation about this topic. But I do want to take I think we come down to 3 minutes, is this really important idea of uploading data and how many of us don't own the data, but that doesn't mean we cannot still be part of the process of educating our collaborators and users. And just, I'd like to get your thoughts on how we can make this happen and things that we can do to contribute to it.
Yeah, I think at first of all, the most efficient way to do or effective way to do is like a cryo-EM community. The journal requires, if you publish volume EM data, you have to deposit in whatever place. If as a whole community, we can give journal those kind of influence, then we don't have an issue.
But what about with your on the ground local users? Like
The local, that's the issue. Like I said, we have many users. So then we gave data to them. We wrote the method. We don't know when the paper will be published. And also, it is who's going to deposit data, they or us. So this is a lot of work we need to work on. So far, we have generated many of the data. We only deposited one in impact.
Michelle,
Yeah, sorry, I moved around.
I was wondering where.
I want to push back a smidge on this idea of data ownership, because in the vast majority of cases, for the data that is collected I would say at most facilities, not all and not all cases, that data is not owned by the researcher either, right?
Publicly
It is a
funded.
public resource.
It is from the university.
Right. Well, it's the university, but it's funded by a public grant.
Very good
And
point.
public grants all, as far as I'm aware, generally speaking, have a requirement for you to make that resource available. Just like a reagent, if you develop a reagent, a new cell line, all of those things. It's the same thing with data. So I do want to try to move away from this idea of data owner, right, being the researcher or the facility or whatever, because I think that that's a bit harmful sometimes, and it allows people to be like mine, and not want to upload it. But that's not actually the situation.
Michelle, I'm so glad that you brought that up, because I think that you touched on a very important point that is actually being required now in some places. So I think it's really important for us as facilities to remind the users of this very important detail, and then identify things that we can do to facilitate this, so they can find resources where they upload stuff, how they're going to go through the process. So things that we can do to help them. I didn't get a chance to.
Yeah, and somebody had to help us how to do it, because we don't know.
So we help them, but we need someone to help us. Rob.
Wouldn't be a very simple solution to have a collaboration contract. They want to use your resources, your support, because they want to have nice images. They say, okay, we can do this. And that also means that whatever comes out of this needs to be in a repository. We can help you find out which repository you agree with this. You sign here, we sign here. If they don't comply, then they won't be able to do their next job with you because they're not adhering to the ground rules that you laid out.
Go ahead.
I really don't think that's the issue about the user doesn't allow us to do it. It's just follow up. I think it's the thing is you know, when we give the data to them, when the paper published, right, that will take maybe 2 years, 3 years later. Then who's going to follow up to put this deposit? If we have a guideline, you know, as a community, that's what we can do. You know, if Vladimir and they were supposed to be follow this guideline to deposit here, then we can give that to user and saying that's what you should do when you publish the paper. In the meantime, deposit this one to that.
But I think what we need to do, the facilities need to do, and working in a country where we all can do recommendations, never impose. Sweden is like that, it's not so simple. But you have to educate, we have to educate them. First, we need to be educated. Now we have managed in Sweden, the same as Germany and France, to mobilize national, because that's important as well. That's included then all the universities. And then the idea will be that nationally will be a way how we will managing the data, including all the data to the repository and before the repository in the moment that you still analyze and processing, because here we need to know differentiated storage and repository and for how long you have to have it accessible to analyze. And we have to, our idea now, once we have that set up, we go with Omero, Protocols.io, and not to remember now the other project, that we will make it together. And once the users start to load in, we do it now locally in our facility, loading the images go transfer directly to Omero. They have to annotate in the normal way they have. And then after, what we want to pretend to do with that is to educate them and to understand, to show them the tools they can have
Education.
The tools they have there.
I want to be
do
mindful.
it.
I think we're done, right? I'm cutting into your coffee break. So it really sounds like there's a lot of discussion on this topic of how to facilitate this, and a lot also around ways to support career development and paths for people working in this field at different levels. So thank all three of you very much for your contributions.